This award is in honor of the first EMBL Director General, John Kendrew. It was launched in 2007 by Konrad Müller on behalf of the EMBL pensioners. And since 2010, is sponsored by Roland Becker, which is a friend of EMBL and private philanthropist. The prize consists of a cash prize of 10,000 euros and a medal, and is given for excellence in science or science communication after leaving EMBL. The prize this year goes to Giorgia Guglielmi for science communication, and it is special because it's only the third time that this prize is given for this subject, communication. So she'll be introduced by former Director General Iain Mattaj. So please, Iain, the stage is yours.
Thank you very much, Fátima, and hi to everyone. I was told that I should start this, even though this is really about Julia, by telling you who I am. I just saw lots of pictures of myself, so I imagine many of you already know this. But I'll give you a little bit of background. So I started my scientific career doing a PhD in Leeds University in England in the 70s, from 74 to 78. And then I did 2 postdoctoral fellowships in Basel in Switzerland between 1979 and 1985. The first was very, very unsuccessful. I was attempting to characterize a gene promoter which was inducible by estrogen. And it essentially, it not only didn't work, but I discovered that quite a lot of the data on which it was based had been faked by previous members of the lab. So this was not a very good start to postdoctoral life. But I changed labs and joined the lab of Eddie de Robertis, who was at the Biozentrum in Basel at the university. And started with him working on the characterization of the genes encoding the USNRNAs, which are involved, among other things, in pre-messenger RNA splicing. And I worked on that topic using xenopus as an organism, and then went on to study the assembly of USNRNP particles in which the USNRNAs act. So this got me interested in nucleocytoplasmic transport, and I was already interested in transcription. At the end of the postdoc, I became a group leader at EMBL, and then went on to be, in order, the head of one of the EMBL units, the scientific director of EMBL, and then director general of EMBL. The entire period spanned from 1985 till the end of 2018. So you've all guessed that I didn't do it in the fixed term of 9 years. This went on for much, much longer than that. And I was one of the lucky people who could stay at EMBL and continuously have new challenges. The science started at EMBL as a continuation of my postdoctoral work in Eddie de Robertis' group. Eddie's group, in parallel to my work on the USNRNAs, had cloned the first vertebrate homeobox gene, again from xenopus. And Eddie was very interested in developmental biology, actually, as Julia was during her PhD and afterwards, and wanted to work on the homeobox genes because this was his way into developmental biology. And so I could take my entire project with me and start work at EMBL working on this. And I continued to work both on snRNA gene promoters and nucleocytoplasmic transport because that was involved. The assembly of an RNP involves making an RNA in the nucleus, proteins in the cytoplasm, and there has to be transport till everything gets together. And the way this happened in the case of the USNRNPs was particularly interesting. And so that was a very successful theme of work, which I continued throughout the time I was at EMBL as it led me into more and more new fields. Eventually, my lab had moved it totally from, if you like, basic gene expression into cell biology, and I worked on nucleocytoplasmic transport and the role of the RAN GTPase, finally working on the roles of RAN in mitosis at the end of the time. Since the beginning of 2019, after I left EMBL, I have become the director of a new institute whose main building is now behind me, called the Human Technopole in Milan, in Italy. Human Technopole is a government-funded foundation whose goal is to set up a research institute working on human health and well-being, but also providing access to Italian scientists to advanced research infrastructures. So the preparation, and you can hear that this bears quite a bit of resemblance to EMBL. And so my background in EMBL has been very useful in getting started here. Human Technopole has started very well. We have recruited excellent researchers to come to work here. In fact, earlier today, I persuaded another head of research center, the equivalent of head of unit, to join Human Technopole. And so another person will be leaving Cambridge to come to Milan to head up one of our research units. So this is all working well. We are building laboratories because the Palazzo Italia building, which is behind me, is a dry lab and office building, and we need to build laboratories. And that, as you can easily imagine, with Italian and European bureaucracy and tendering and building processes, is a delight without end. Particularly the without end part is true. So with that background, I'd like to go on to today's task. My task today, and it's a pleasure, is to introduce this year's John Kendrew Award winner, Giorgia Guglielmi, who actually also gives me the occasion to practice 3 of the 4 different ways that you can pronounce a G in the Italian language. So thank you, Giorgia, and your parents, I guess. Giorgia was a PhD student from 2011 until mid-2016 in the laboratory of Stefano de Renzis in developmental biology at EMBL Heidelberg. Her project took the technology of optogenetics, which was proving extremely useful in the context of neuroscience and activation of neurons to fire, and readjusted the technology so that it could be used to tackle problems in cell biology through activating signaling in cells, in particular parts of an organism, through the light of, through the action of laser light. She then went to the US, where she did a science writing course, which was located at MIT for 1 academic year, before beginning to write for various different scientific journals, initially doing internships with both Science and Nature before becoming a freelance writer. I find the topics which she has written about, both during her PhD at EMBL and since leaving EMBL, very, very interesting ones. She talks about various examples of the use and support of pseudoscience and the damaging effects that fake reports, if you like, or reports about fake science can really have a damaging effect in various aspects of society. She talks a lot in her more recent work about the diversity, or perhaps more precisely said, the lack of diversity in the life sciences in general, both in terms of people and in the way that that limits the choice of topics for research in the life sciences. Her writing has been acknowledged. She won a Travel Award this year and was nominated for the American Association for the Advancement of Science Kavli Science Journalism Award, which has not yet been selected. She still has a chance to win that prize. And now she's been chosen from a group of excellent candidates as this year's EMBL Alumni Association John Kendrew Award winner. So please join me in congratulating Giulia as a very worthy winner of the John Kendrew Award.
Thank you so much. This is the award, the certificate, and this is this wonderful medal. My name on the other side.
Fantastic, Giorgia.
Thank you.
So, please tell us your story.
Yeah, thank you so much, Iain, for the kind introduction and for pronouncing impeccably my surname. However, you made a slight mistake of my name, because my name is Giorgia, it's not Giulia.
Oh my God, I'm sorry, I knew that. In fact, there are 4 G's in your name. And
yeah, I know, too many G's. But yeah, don't worry, everyone keeps calling me Giulia, so maybe that should have been my name. I don't know. But anyway, I will just share my screen for showing you a couple of slides that I prepared for today. Okay, so, first of all, I would like to thank you, of course, the people who nominated me for the John Kendrew Award and the committee, the award committee for selecting me for this award for this year. And most of all, I want to thank you all for, I know you're a lot of people, and I want to thank you all for sitting in front of your computer on a summer Friday. So I know it's not easy, but so I'm going to keep it brief and informal. I always thought that my talk should cover what I did during my time at EMBL, and also the science communication for which I won the John Kendrew Award. Now, I don't know if the 3 stories that I'm going to tell you about today are those that got me the award, but they are definitely the stories that matter the most to me, and I hope you'll also find them important and worth covering. So now let's start from where everything began, EMBL. So this is a pretty accurate representation of my first year as a PhD student. Of course, I was a bit taller when I started at EMBL in 2011, but like in this picture, I was pretty much clueless. Before joining EMBL, I used to work with bacteria, so with single cells that behave in a rather simple way. With all due respects to microbes and microbiologists. And instead in the Renzis lab, I had to deal with, fruit-fried embryos made of thousands of cells that constantly exchange information, influence each other, like you see here in this movie. They move, they shrink in size, they divide, it's a complete mess. And much of what we knew or know about the biological process that drive development comes from turning on and off specific genes in the embryo and then watching for the effect. But most of the time you have to wait, sometimes several hours in the best case before looking at the effects of this perturbation. So you cannot really tell what exactly went wrong, when and where it went wrong. So when I joined Stefano's lab, my dream was to have a remote controller to perturb the activity of single cells and watch for the effects immediately. And I thought that would allow me to understand when and where specific cell behaviors were needed during development. And to do so, as Iain said, I decided to use a technique called optogenetics, which had allowed neuroscientists to control the functions or the behavior of cells with a flash of light. And because many of the movements that I just showed you before in the previous slide are driven by cell contracting and shrinking in size, I decided to control cell contraction. So it took a couple of years and many failed experiments, but eventually I succeeded in developing a tool that allowed me to control cell contractility with light. And so in the system I developed, when lights are off, cell can contract. But when I illuminate them with blue light, they cannot contract anymore. And applied this to fruit fly embryos to understand how they develop their shape. And here you can see this nice embryo developing in the dark, and it just does its own thing. Cells on the underside of the embryo at some point start moving inwards. They fold into the embryo where they eventually will give rise to muscle. But when lights are on, this whole process is blocked, as you can see in this other video. Now, the cool thing about this system is that I could direct the light only to certain areas of the embryo and prevent cells from contracting only in a particular area at a particular time point. For example, here, in the blue box, the illuminated cells will not contract while their neighbors, which stay in the dark, would, and that's how it looks in real life under the microscope. By changing this light pattern, I could really study how this inward folding process that I showed you before works. I could study where the contracting cells should be positioned for the process to happen, how many cells should contract, what kind of sort of shape they had to assume. And to my knowledge, before then, no one had ever used optogenetics to interrogate how cells behave and interact with each other in a whole organism during development. And so this study was sort of the proof of principle that this could be done. And I'm proud to see now that 5 years on, this approach has been used in Stefano's lab and in other labs around the world to ask their favorite questions about development. But to be completely honest, that was not the biggest discovery that I made at EMBL. The biggest discovery I made was that I could actually combine my background, my scientific background, with a lifelong passion of mine, which was writing. And by talking with people in the EMBL communication office and by actually talking to them and see what they were doing, I realized that I could write about science and actually make a living out of it. So I started to collaborate with the EMBL communication office. I wrote some articles for EMBL, etc. And that's really when I decided that I wanted to switch career and become a science communicator. And so in 2016, it was just a few months after I got my PhD from EMBL, I moved to Cambridge, Massachusetts to attend a graduate program in science writing. And today, three years after graduating from the MIT, I'm a freelance journalist covering the life sciences, biomedicine, and science policy. Now I know probably this audience is full of scientists. I don't know how many of you are familiar with science journalism. Probably some believe that the duty of a science journalist is to convey information in a way that's compelling and clear to the broad public, which is true, but our job does not stop there. Science journalists try to capture the beauty of scientific research, but also its shortcomings and its biases. We ask critical questions about statistics, sample size, research design, but also about ethics, the impact of scientific findings on society. We investigate research misconduct, sexual harassment, racism, discrimination in science. And I think that's a very important duty of science journalists, try to heal the disconnect between public opinion and the scientific consensus on many topics, from vaccine safety to evolution to climate change. And that's because people's decisions at the end of the day are influenced by what they read, what they see or hear in TV. And the media can end up feeding readers with misinformation, even fake news that influence at the end how people perceive science and its value. And this really brings me to the first of the stories that I wanted to share with you. and that Iain, I think, mentioned earlier. So this was actually my thesis for the MIT graduate program in science writing, and it analyzed how misinformation has led to the death of hundreds of thousands of olive trees in southern Italy. And let me tell you just a few things about this. So in 2013, a deadly microbe started to infect olive trees in Salento, which is Italy's most southeastern region. And the microbe is spread by insects, and it started to kill trees, compromising the production of olive oil, which is very important for the region. And since there is no way to cure sick plants, Italian authorities ordered emergency measures to contain the disease, which included removing the infected trees and using pesticides against insect vectors. But in Salento, these measures sparked intense public opposition, mostly because some shoddy media reports, which were echoed by public figures like football players and singers, argued that the olive tree emergency had been made up to help biotech companies to sell genetically modified olive trees in Salento. And so under immense public pressure, eventually an Italian court halted the containment measures and accused the scientists who detected the microbe as having caused the problem in the first place. So this was a very, very, sad and messy story. And for this, I interviewed, everyone from conspiracy theories to government officials, scientists, and local farmers to reconstruct really what happened, what caused the misinformation to spread, and also help the public to have a clear understanding of what the issue was and the science behind it. And so I wrote about this in 2017, and since then the criminal charges against the scientists have been dropped. But still today, there isn't a clear plan to contain the epidemic in Apulia and Salento, and the public resistance has fueled the spread of the disease, which has now killed, I think, more than 1 million olive trees with catastrophic economic consequences for the region. The second story I want to tell you about, it deals with science and social justice, if we want to say so, and how a growing number of scientists, many of whom come from groups that are underrepresented in the research workforce, are trying to decolonize genomics, meaning they're trying to make genomics more inclusive by consulting and involving indigenous and other marginalized population in their projects. The problem is that genome studies, as you can see here, have historically focused on people of European descent. As of 2018, only 22% of people in genome-wide association study were non-European ancestry. And the reason behind this lack of diversity in genome research are many, but I think definitely they include a past of abuses of indigenous populations by Western scientists. And now this handful of scientists are trying to stop this cycle of disengagement. They're publishing ethical recommendations, they're launching training programs for indigenous people, and they're really leading the way in working with minority groups in genome research. And I was talking to many indigenous and non-indigenous researchers. And, they were all telling me that, these efforts are desperately needed. This is Keolo Fox. He's a native Hawaiian researcher at the University of California, San Diego. And really, I couldn't agree more with what they were telling me. I really think we need to mitigate, the legacy of Western culture in research. And it's not just about increasing the number of scientists from racial minorities, but also, trying to respect the traditional knowledge, the local philosophy, the traditions of indigenous population that Western scientists have overlooked and, often rejected. And the third and last story that I want to tell you about is about diversity in science. This is something that I wrote last year. It's a profile of Cassandra Extawar. She's a Harvard professor who appended the leading theory of how most animals generate the precursors of eggs and sperm, and she cracked a long-standing question about the diversity of insect eggs. And she did all this while facing racism and discrimination as a gay Black woman in science. I remember meeting her when she was giving a blue seminar in the developmental biology unit, and I was really impressed by her science. But besides being an accomplished scientist, she's also a professional soprano and really an advocate for diversity in academia. And my hope is to see more and more people like Sandra Exter in science and in science journalism. There's a question that I often ask myself, and is how can we boost diversity, inclusion and representation in science and science journalism. And I think as a science journalist, my duty is really to produce work that makes an impact, like on public understanding of science, that's for sure, but also on public understanding of who counts as a scientist and what kind of science is worth covering. And that's why in my coverage, I try to include more diverse sources, and I focus on the representation of minority groups in academia, and I try to counteract, the stereotype of a scientist as an old white male. So I care about Nobel Prizes, of course, but I also think that a story about, researchers like Keolo Fox and his colleagues who are making a difference in their local communities could really make a more lasting impact. And I'm really grateful to the John Kendrew Award committee for recognizing this effort. And I'm also grateful to the committee for showing that science journalism that tries to pursue the truth is key and is critically important in this age of fake news and quote unquote alternative facts. And I think another important thing that I want to mention before closing is that the decision to give this year's prize to someone like me who left academia sends a really important message, that the scientific community is starting to recognize that PhD graduates are qualified for a variety of careers that don't stop in academia, and they're able to have a positive impact on both science and society, even if they no longer work in academia or at the lab bench. So thank you once again to the committee for this honor. And before taking questions, let me thank the people who made the stories that I talked to you about and all my other stories able to see in the light. So my editors, teachers and mentors who taught me everything that I know about science journalism, and they helped me to find my voice as a writer, and my family and friends for their support, their unyielding support, because without them, of course, I wouldn't be even here today. So with this, I am gonna stop sharing my screen. Well, if you want to keep updated with my story, just follow me on Instagram or Twitter. I also have a website if you want to reach out. If you don't manage to ask a question today, just use the comment, the message section in my website, and I would be happy to answer to any question you have. But thank you.